We left off here, we were describing water reabsorption. And just to go back here, one slide back, just to review the proportion of water being reabsorbed in different parts of the tubule. So the most water reabsorption happens in the proximal tubule. We describe this as being unregulated in that it happens freely as long as there's a solid gradient. Water can follow by osmosis. About 20% of that happens in the distal part of the tubule. This is under the reabsorption regulated by ADH. So in the distal tubule and the collecting duct, we have more of a regulated reabsorption, but it's to a lesser extent, right? About 30% collectively regulated with the hormone ADH. So let's talk about how that regulation happens. Why doesn't water get reabsorbed freely in these later parts of the tubule? And that is dependent on the osmotic gradient that we created through the loop of Henle. It is also because the permeability to water in the distal tubule and collecting duct are dependent on aquaporins. So water reabsorption doesn't happen freely across the membrane, but it happens through these designated water channels called aquaporins. There are two types. Aquaporin 3 is on the basolateral membrane, and it is always present, whereas aquaporin 2 is on the apical membrane, and it is only present when it is stimulated by production in the blood, as through the mechanism that we'll talk about. So basically upregulation because of receptor binding of ADH, and then we can insert more of those aquaporin 2s into the apical membrane to allow for the water to get into the cell, and then it can be reabsorbed according to the basolateral membrane, which has the aquaporin 3. So two different sides of the cell, and we can have water absorption with both of those aquaporins. So whenever we think about the late distal tubule and collecting duct, they're usually impermeable to water, which means that water cannot leave freely. There's no free reabsorption, and more water is going to be excreted into the urine. So that simply means that our urine is going to be concentrated through the reabsorption of water in these parts of the tubule, which is what is going to prevent us from excreting pure water. What we excrete is really the dissolved solutes in a very concentrated filtrate, which is what we know as urine. Most of that water gets reabsorbed and reclaimed through these two mechanisms with aquaporin and ADH. So ADH can stimulate the insertion of these channels into the apical surface, as we just mentioned, the aquaporin 2 channels. Water can be reabsorbed into the cell, right, by osmosis, according to the gradient that already exists. And then the concentration of urine happens through this mechanism. So the urine gets concentrated up to about 1,400 milliosmoles. Instead of us excreting urine that is extremely diluted, excuse me, not diluted, diluted at about 100 or even 200 milliosmoles, we want to pull all that water back into the body, and we want to excrete only the minimum water necessary, which is that obligatory water loss. So we want to excrete solutes and the least amount of water possible, which is why we reclaim and we absorb that water in a regulated way in the distal tubule. And so the maximum amount of water that we reabsorb is dependent on the loop of Henle. This is where that medullary osmotic gradient is created. So if we have a really long loop of Henle, we can really concentrate the urine extensively. If you look at the loops of Henle of certain species and certain animals like camels or desert rats, they have very, very, very long loops of Henle because they have the ability to create extremely concentrated urine. Their maximum urine concentration is probably much higher than ours, this 1,400 milliosmoles. And so that loop of Henle length correlates to the kidney's ability to conserve water, to reabsorb more water, and to eliminate as little water as possible, right, with the solutes. So here is sort of a visual example of that. So here's our tubular fluid as it comes to the collecting duct, which is the final part of the loop of Henle, the distal tubule and the collecting duct. They're impermeable to water, typically. And so this is what would happen if we didn't reabsorb any of that water. Our urine filtrate would be high volume, low osmolarity. So we'd be excreting a lot of urine, which would be a lot of water, and have a low osmolarity with very high water, very little solute in terms of that proportion. Now, in terms of our kidneys and what is needed is we need to conserve water as a part of our function of evolution in times when we cannot have access to water. And so the distal tubule collecting duct have the ability to insert these aquaporins so that we can reabsorb more water. And as we do that, we concentrate the urine. So the urine osmolarity goes from very low up here at about 300 milliosmoles. And as we reabsorb, reclaim most of that water, we see that the final urine filtrate has a very low urine volume, but a high urine osmolarity at about 1400 milliosmoles. So notice how the length of the loop creates the space and time to reabsorb and reclaim water. If we had a very short loop of Henle, we would not be able to reabsorb as much water, and our urine osmolarity would be much higher, or excuse me, much lower. Volume would be higher and osmolarity would be lower. But if we had a very long loop of Henle, again, like some of those other animals I mentioned, we would see that we would have even longer length, more space to reabsorb more water, and have an even higher osmolarity and even lower volume. Okay, this brings us to talking about that obligatory water loss. It's the minimum amount of water that must be excreted from the kidneys per day. We describe that as being about 440 milliliters, and the osmolarity of that urine being about 1400 milliosmoles. So the urine is really to secrete the waste, the solute waste. That's what we're really trying to eliminate. We're not trying to rid the body of water, of pure water, which is needed for hydration, which is needed to maintain our mean arterial pressure, our blood pressure. We don't want to eliminate just fluid just for the sake of it. We want to eliminate the waste, the solutes that have been filtered from our blood, and any excess ions, toxins that we would like to eliminate, and not so much the water. So the minimum water, 440 milliliters. Of course, if you're more hydrated, if you have you've ingested a lot of water in a day, your minimum water loss might be higher than this, but for the normal state, this is the minimum amount. And so that is what is necessary just to get rid of the solutes and those waste particles. All right, let's take a brief pause here. So our third learning objective to explain the control of water balance and osmolarity by antidiuretic hormone. Now, looking at the effects of ADH on water reabsorption, we just described how it can upregulate the insertion of aquaporin 2 channels. And so it basically regulates the permeability of the distal part of our tubules, the distal convoluted tubule and the collecting duct. And it helps us to convert our urine osmolarity from 100 milliosmoles, which is the state of the filtrate coming out of that ascending limb of the loop of Henle, if you remember that. As soon as it goes down into the loop of Henle and comes back up, it's very, very low, even lower than it was in the proximal tubule at 300. So now it's 100, right? Very diluted. And we want to concentrate it in order to preserve some of that water loss. So we're going to insert those aquaporin 2 channels, reabsorb that water, and it goes up to about 1400 milliosmoles. And the whole role of this hormone is to prevent diuresis, right? If you look at the name ADH, antidiuretic, it's going against diuresis or preventing that diuretic function of the kidney. Diuretic means that it's going to eliminate water, And there are a class of drugs called diuretics that do that. This can be important in states of hypertension. A lot of the hypertension medications, some of the heart failure medications are actually diuretics. They help us to lose some of that water so that the blood pressure can drop. In this instance, this hormone is working against that. It's trying to conserve our water. And so it's going to insert these channels so that we can reclaim it by reabsorbing it in the distal tubule. Let's look at the mechanism of how that happens. So ADH is a hormone. It actually is secreted in the posterior pituitary. It makes its way into the blood, into the plasma, and then it circulates until it gets to the distal part of the kidneys, the epithelial cells in the distal kidneys. And so what it does is it binds to receptors here. There are specific ADH receptors on the basolateral surface of these epithelial cells. It crosses the membrane, binds to these receptors, and then it stimulates a second messenger cascade by activating a G protein, activating an amplifier enzyme, and then creating its second messenger. In this case, it is cyclic AMP. That results in turning on a protein kinase A in this case, and then that essentially makes and transports aquaporin 2 channels into the apical surface of our epithelial cells in this part of the tubule. So this is an epithelial cell in both parts of the tubule, both the late distal collecting, or excuse me, distal convoluted tubule and the larger collecting duct. So once we upregulate, and upregulate just means that we're turning on the genes that make more of these channels, and then we also transport them to be inserted into the apical surface of the membrane. Now we can have water being reabsorbed across the apical surface. And then remember the other aquaporin 2, aquaporin channels that we described, the aquaporin 3 channels, they are always in the basolateral surface. So they're always present in this basal surface. All we need to do is increase the aquaporin 2 so that water can get into the cell, and then it can freely move across the basal surface and then be reabsorbed into the plasma or the blood. All right. So just to recap some of those points here, the origin of ADH happens from the posterior pituitary. Remember, it is made in the hypothalamus, but is stored and released in the posterior pituitary. If you think back to some of our endocrine lectures last semester, it is basically being stored as it is made from the nuclei in the hypothalamus. It is released through those neuroendocrine hormones and then is stored in the posterior pituitary and released upon trigger from the hypothalamus. It is also going to circulate and be triggered by osmoreceptors in the brain. These are called OVLT receptors, organum vasculosum laminae terminalis. Very fancy word, but it is one of the areas in our brain that can sense and detect our osmolarity of plasma. And so it will determine if we need to secrete more of that ADH or less of that ADH, depending on the osmolarity of our blood. Okay. And it is surrounding a part of the brain that is not exposed to the blood-brain barrier so that it can freely detect the blood and it can freely detect the osmolarity of the blood and it can trigger the release of that ADH from the posterior pituitary. It is also going to be affected by baroreceptors. So baroreceptors in areas like um, the common carotid receptors that are in the neck, they can detect the pressure of blood flowing past those mechanical receptors, and they can detect any drop in blood pressure or blood volume, and then they can also, in response, secrete ADH in order to preserve water and raise that blood pressure. So remember, decreased baroreceptor activity means that the pressure in the blood passing these receptors is low. And so the response to that is to secrete more ADH, to reabsorb more water, to potentially correct that mean arterial pressure drop, and then that can correct and restore the blood pressure and blood volume.